I am going to talk about rotavirus A, particularly the G3 and G14 types, as well as rotavirus B, the newly discovered virus, testing and review of our laboratory cases. So briefly, I know many of those speakers talked about rotavirus before. But again, briefly, these are non-enveloped, double-strand RNA viruses under the family of Reobridia. There are 11 RNA segments encoding 6 structural proteins and 6 non-structural proteins. So based on those VP6 identity, which is the structural protein, rotavirus are classified into 9 groups. And rotavirus group A is the leading cause of the diarrhea in humans and animals, and also in foals. So, and group A is further classified into G types and P types based on the nucleotide sequence of VP7 and VP4 protein. So this is a very traditional classification method. I know that there are lots of studies on the way for a new classification method based on the whole genome sequence rather than the only structural proteins. So, so there are 27 G types also and 35 P types. So as you can see, there are lots of variabilities within the group A. And the one reason is that this is an RNA virus. The mutation rate is very high. Recombination rare certains can happen very much in this type of RNA viruses. So within those rotavirus A, group A also, there are several types affects the horses, particularly G3 and G14, the most common types. According to a recent paper, Dr. Balasuria, actually he found only G3 and G14 are present in central Kentucky area. So rotavirus B never been detected in foals. It has been detected in some other animal species, pigs, calves, and humans, but not in horses, except in one case where recently in Germany they just screened some fecal samples for real-time PCR, and they detected only one case without any further looking at if there is an association between this one positive case and the diarrhea. So this is the sort of the timetable for this idiopathic foal diarrhea outbreak 2021 diagnostic efforts for rotavirus etiology. So this figure has been modified from Dr. Craig Carter, and I appreciate for putting this all together, Dr. Carter. Everything is start when Dr. Adam approached us that there is a problem in the industry, diarrhea problem. I think there was also another problem, a liver problem at the same time. So I really appreciate your effort in initiating this project altogether. Then February 25th, I remember the phone call from Dr. Slovis. He called me, he said that there are diarrhea problem. He got some samples, he tested those for a number of disease agents, rotavirus A, coronavirus, C. C. diff, and they were all negative except that 3 cases, there was Clostridium perfringens type A positive. And I told him, well, why don't you just send those 2 samples to us and let's just take a look in our lab and see what we are getting. And basically, we did similar testing, we got the same result. So we confirmed that pretty much, nothing, because we already know that Clostridium perfringens type A can be found in healthy foals as well. So at that point, it was inconclusive to us. And then because of the nature of this, spread of the disease at the time, we decided to send those samples for electron microscopy analysis. So that we used to have an electron microscopy unit in the college, but so there is only one in, I think, in engineering department, and I called them, hey, we got the situation here. I'd like to send those samples to you guys. Can you look? And they say, well, what I understand from that, they were sort of avoiding the animal or human specimen. So medical school also send their sample to somewhere else. So we send all samples to Texas A&M first, and then the report came back, one of them was positive for rotavirus. So again, electron microscope cannot differentiate if it's rotavirus A, B, or C, or whatever. So it just tells this is rotavirus. And then unfortunately, the camera system was broken. And I just need the picture really badly at the time, because sometimes one picture is worth of 1,000 words. So, and then I sent the similar samples to University of Missouri, and I got a really nice picture from them, and I'll show you in a minute. And then Dr. Lee forwarded those specimens collected, particularly from Dr. Erdal and others, and sent to South Dakota State University. And then we got a response from Dr. House from South Dakota saying that 2 pooled fecal samples contain lots of new rotavirus type B. And then eventually we developed a real-time PCR assay that can detect this particular virus, both our lab and Dr. Lee's lab. And then the test was offered to clients by our lab and eventually. And finally, those publications, the findings of publication of these findings published by Dr. Lee Group, very elegantly written paper and published in the journal as he mentioned. So, and this is the picture of the transmission electron microscope of rotavirus B particles. So these are particles contain lots of intermediate replicates. So you see a lot of particles here, and both of those electron microscope experts from both TVM and the University of Missouri told me that there are lots of replication going on in this pulse. And lots of replicative intermediates, they are called. And as you can see, single and clusters of these non-enveloped electron-lucent particles with electron dense characteristic of rotavirus particles. So in this figure, I'm showing you some real-time PCR testing for rotavirus A, and see what the situation is look like right now at the University of Kentucky Veterinary Diagnostic Lab. And you see the years, and also the total numbers, and you see that there are positivity in pretty much every year since 2018. So this is the year we started with this multiplex equine diarrhea panel. So we can look at our data retrospectively and see how many samples we processed, tested, and how many positive we have. So as you can see, there are lots of positivity on those over the years for rotavirus A, which is really expected. So it's a common disease. And these are, by the way, from the necropsy cases. I did not include those routine cases because necropsy case usually better description of the clinical history on the submission board. And when we look at those, the genotyping for G3 and G14 on some of those samples. And as you can see, we got 22 samples. I look at 2018 and some of the 2019 cases. And as you can see, the CT values, 20s, usually a few 30s, 15, some of them. So fairly, strong positivity. And then when you look at the genotyping, most of them are G14, those 2 years. So we got 4 G3s and 14 G14s, and one case at both G3 and G14. And then when you look at the ages and of those cases, you see there are several weeks of falls, few months of falls, and also usually the sex distribution. And when you look at the diagnosis, these are the pathology diagnoses. You see most of them have some sort of, intestinal problems. And it's interesting that someone has, I think you mentioned earlier that there may be some association between this perforation and gastroduodenal ulceration. And I do see that in many of those cases actually indeed have this type of problems. But again, rotavirus A may not be, the killing factor in these cases. The foals may have died somewhere else, most likely perforation on those cases. But it will be interesting to actually establish a relationship between, if there is a relationship between this virus and gastrointestinal ulceration or perforation. And this is now showing that, this picture showing a multiplex real-time PCR testing for rotavirus B at diagnostic lab. So again, this test was developed very recently, just a couple of months ago. So in the bottom, you see 2021 results. We tested 122 cases, necropsy cases, and we got 115 was negative, and then 7 positive cases on those necropsy cases. So in our lab, we have archived samples. So we do keep the RNA extract from early years samples. And we keep them in -80. I did screen some of those samples, and as you can see, starting from 2017, all those tested samples are negative for rotavirus B, suggesting that the virus did not exist, at least during this period of time. It could be possibly that maybe it was here before, but at least starting from 2017, 18, 19, 20, I could not find any positive samples in falls during this period of time. And when we look at further for those 7 cases, as you can see that those rotavirus B cases usually be detected during February and March, as was discussed before also. And CT values, again, usually you see the 20s. And again, age, again, confirming earlier results that most of those cases like just a few days old, unlike rotavirus A. So usually like 2 days or 3 days or 5 days. So, and the clinical history, again, confirming that there is almost always there's a diarrhea or dehydration or colic, again, confirming this that there is a rotavirus association with this type of with this cases. So in conclusion, when we look at the equine rotavirus A cases, they are usually several weeks old falls affected. Again, confirming all earlier studies. It's a common pathogen, ubiquitous. It's present every year since we test here. And then also the majority are G14 genotype, confirming Dr. Balasuria's early results. On the other hand, equine rotavirus B is usually less than 1 week old falls are affected, and mostly in February and March so far. Now, of course, we don't know if this will happen next year or next years. And those are interesting questions Dr. Lee raised. Where did they come from? And will it happen again? Of course, there may be some, other host, intermediate host or some maybe maintenance host, possible maybe. Also, probably one need to look at the presence of this virus in older animals, older horses. So maybe because rotavirus B is found in adult humans as well. So maybe this virus is indeed present in older horses. If that's the case, we may see this problem next years as well. Also, as I mentioned earlier, this virus does not detect in archive specimens in earlier years from 2017 to 2020. So with that, I would like to acknowledge the practitioners. I really appreciate it. The staff and faculty, particularly molecular diagnostic, bacteriology, and pathology, for the excellent work. I really appreciate it. Dr. Kader and Dr. Horohov, not only for their leadership, but also their contribution, intellectual contribution. And again, Dr. Adam, for your leadership on this one. And certainly, Dr. Thanks for your excellent work and the publication of initiative of the next generation sequencing and the publication of the entire work. And also, I would like to thank Dr. Solomon and Dr. Farrow from University of Missouri and Texas A&M Diagnostic Lab, respectively. And as well as Dr. House for doing this next generation sequencing from South Dakota State University. With that, I also thank you very much for your attention. And if you have any questions, we'll answer. Thank you.